Hello guys, welcome to Easy Bio. Today's video is going to be a continuation from my first enzymes video. So if you haven't seen that yet, please go and watch that before this one. But first, let's do a recap. Enzymes are biological catalysts. This means that they speed up reactions in your cells. The reason why enzymes are so important is because they speed up cellular reactions. If they didn't do this, you would die. Enzymes have active sites that substrates bind to, and a product is then made afterwards. The active site is specific only to a certain substrate. We call this being complementary to each other. Finally, the enzyme is not changed in any of these reactions and so can be used again. And that's the end of the recap. Let's get into the actual video. Okay, so the focus of today's video is enzyme function and conditions, which basically means the environments enzymes work in. First, let's familiarize ourselves with the term optimum condition. Optimum means the best possible result, and condition, in this case, is the environments the enzymes work in. So optimum condition is the environment an enzyme could produce the best possible results in. If you were to look at a graph, you will be able to figure out the optimum condition by seeing the point where enzyme activity is highest. So yeah, the rate of reaction is the highest it can be. A bacteria called Thermus aquaticus lives in hot springs. The enzymes have an optimum temperature of 70 degrees Celsius. That's crazy compared to the enzymes in our body that have an optimum of 37.5 degrees Celsius. Now let's talk a little bit more about temperature and enzymes. First, let's understand what temperature actually is. We've always heard the word temperature being thrown around, but do you actually know what it means? Temperature is the measure of kinetic energy a molecule has. A simple way to describe kinetic energy is moving energy. It's the energy that moving objects have. So temperature is a measure of how much energy is in a moving object. So, as shown by the graph, as the temperature increases, so does enzyme activity until after the optimum temperature. The graph also indicates where the optimum temperature of the enzyme is, the peak of the graph. Why does this happen though? Well, there's something called collision theory. Basically, it states that in order for a reaction to occur, the molecules have to collide with each other first. When you increase the temperature, the kinetic energy in a molecule also increases. This means that they are moving around a lot more. Remember, kinetic energy is moving energy, which leads to a higher chance of collisions and therefore a higher rate of reaction. The reason why the optimal temperature is perfect is because the kinetic energy in the molecule and substrate are just right for the maximum number of collisions. If you remember from the graph, after the peak, the rate of enzyme activity decreases. That's because the enzymes become denatured. When an enzyme is denatured, the shape of the active site changes, and so is no longer complementary to the substrate. So, remember, are proteins, and proteins are made up of amino acids. Amino acids are joined together by something called peptide bonds to start forming a protein. In high temperatures, these bonds are broken by the intense heat, causing a change in the specific structure of an enzyme. This means that the active site is no longer specific to the substrate, and the substrate can no longer fit. This causes the reaction to stop. Okay, so let's take a look at another factor that affects enzyme activity: pH. Like temperature, increasing the pH increases the rate of reaction until after the optimum pH. As you can see from the graph, different enzymes all have different optimum pHs. And just like temperature, after the optimum pH, the rate of reaction also decreases. The rate of reaction also decreases after the optimum pH because of the same reason as temperature. The enzyme becomes denatured. In pH's case, this is because the environment is either too acidic or too alkaline for the enzyme, and so it becomes denatured. Much like the temperature, denaturation happens because, in this case, the intense acidic or alkaline conditions break the chemical bonds in a protein and cause a change to the specific structure of an enzyme, which means that the substrate can no longer fit to the enzyme and the reaction can no longer occur. Every enzyme has a different working range, but the optimum is where they work the best in. So, if we take pepsin, which is found in our stomach, it has an optimum pH of 2.5, but a working range of between 1 pH 1 to 7. So quite acidic, but that's perfect for our stomach. Going back to the jigsaw analogy in my first video about enzymes, all jigsaws have different pieces and different ways to put them together. And that obviously means that they have different pictures. This applies to enzymes as well. Enzymes all have different arrangements and different bonds. This means that enzymes are also all going to be different from each other in many ways, including optimum pH and temperature. But anyway, that's it. That's the end of enzymes. The next video will be about genetic engineering, so stay tuned for that. Bye!